You know, when you're fighting a deadly infection, getting that first clue is everything. Today, we're going to look at a technique that's been giving doctors that crucial first step for 140 years. It's called the Gram stain.
Okay, so picture this. A patient comes in, they're really sick with some kind of mysterious infection. The clock is absolutely ticking. A doctor has to pick the right antibiotic, but there are literally hundreds of different bacteria it could be. So how do you fight an enemy you can't even see? It's a massive challenge, and you need answers, like now.
So you'd think the first thing they do is some super high-tech genetic sequencing, right? Nope. Incredibly, the first test a lab usually runs is a technique that's been around since the 1880s. It's the Gram stain. And it's that very first, crucial step that gives doctors the clues they desperately need.
I mean, the fact that this test is still around is just remarkable. It was cooked up way back in 1882 by a Danish bacteriologist named Hans Christian Gram. He was just trying to see the bacteria that cause pneumonia in lung tissue. And here we are over 140 years later, and it is still one of the most essential tools in any microbiology lab. That's staying power.
So how does it actually work, right? How can one simple stain basically split the entire bacterial kingdom into two huge groups? Well, the secret. It's all about the cell wall. The key player here is the stuff called peptidoglycan. Just think of it like a chain link fence or a mesh that gives the bacterial cell its strength. And the entire Gram stain process, the whole shebang, hinges on one simple fact. Some bacteria have a really thick mesh wall, and some have a really thin one.
Okay, this is the aha moment. You've got gram-positive bacteria, which have this super thick, absorbent layer of that peptidoglycan. Let's call it a thick mesh sweater. It just soaks things up. Then you have gram-negative bacteria. They've got a really thin layer, but, and this is key, it's covered by an outer fatty membrane. Think of it like a thin raincoat over a t-shirt. And that one single difference is what makes this whole thing possible.
So, because of that difference, we get a really obvious visual result. The mesh sweater on the gram-positive bacteria, it soaks up that first dye and just hangs onto it for dear life, turning a deep purple. But the thin raincoat on the gram-negative guys, it can't hold that first dye when it gets washed away. But what it can do is pick up the second dye, which is a pinkish red color. So, purple or pink, that is the first massive clue.
So how do we actually get the bacteria to show us their true colors? Well, it's this beautifully simple four-step process. I like to think of it as a chemical interrogation, designed to make them confess who they are.
The whole process is just elegant. First, you flood the sample with a dye called crystal violet. Bam! Everything turns purple. Next up, you add something called Gram's iodine. This basically acts like a glue, helping the dye stick. It forms this big complex with the violet dye and kind of locks it in place. Then comes the most important step, the alcohol wash. This is the decolorizer. It completely strips the purple color from the gram-negative bacteria. But it just can't get through that thick wall of the gram-positive guys. So now you've got purple bacteria and clear bacteria. And finally, you add a pink counterstain, which colors all those now clear gram-negative bacteria so you can see them again. And I gotta tell you, that decolorizing step, it's everything. As research from places like StatPearls points out, the timing has to be perfect. If you do it for too long, you can wash the stain out of all the bacteria, which means your gram-positive ones will look like they're gram-negative, a false negative. But if you do it for too short a time, the gram-negative bacteria might hold on to that purple stain. It's a step that takes some serious skill and precision.
All right, so the staining is done. Now the real detective work begins. A lab tech peers through the microscope, and they're not just looking for colors. They're trying to decode a whole visual language of shapes and patterns.
So what are they looking for? Two main things, color and shape. The color, of course, tells them if it's gram-positive, that's purple, or gram-negative, pink. Then the shape gives them the next piece of the puzzle. Are they looking at little rods called bacilli, or are they spheres, which we call cocci?
And these clues, they are so powerful. Let's say the lab tech sees purple spheres all bunched up like grapes. Right away, they're thinking staphylococcus, like a staph infection. What if they see purple spheres in a chain? That screams streptococcus, the bug that causes strep throat. Pink rods? High chance it's E. coli. And pink spheres in pairs could be Neisseria, which can cause meningitis. You see, in just a few minutes, that huge list of possible suspects gets way, way smaller.
So all of this incredible detective work leads right back to that sick patient we talked about. The Gram stain isn't just some neat lab trick. It is a critical, frontline tool for choosing the right weapon to fight that infection.
So, why does purple versus pink matter so much?
because that difference in the cell wall, the sweater versus the raincoat, completely changes which antibiotics will work. See, drugs like penicillin are designed to attack that thick mesh sweater of gram-positive bacteria directly. But that same drug just bounces off the raincoat of the gram-negative ones. They've got this outer armor, so they need a totally different kind of antibiotic. The gram stain gives doctors this intel immediately. They can pick an antibiotic that's likely to work right away, instead of waiting a day or two for a full culture to grow. And for a patient with something serious like meningitis, that speed can literally be the difference between life and death.
Now, of course, the test isn't perfect. It's not a silver bullet. It's really a preliminary result, not the final definitive answer. And it doesn't work on every single kind of bacteria. You have some like mycoplasma that don't even have a cell wall, so they just won't show up at all. But as that first critical step, its value is just immense.
It's really kind of a fascinating paradox when you think about it. We live in this age of gene sequencing and all these advanced diagnostics, and yet a simple 4-step stain from the 1880s is still one of the most important tests in a hospital. It's just such a powerful reminder that sometimes the most elegant and simple ideas are the ones that last the longest. It really makes you wonder, what does the staying power of the gram stain teach us about what makes an idea not just brilliant, but truly timeless?